Right. Thank you, Candice, for a very nice talk. If you could switch off your slide, then I'd like to ask all the participants in this session to put on their video for the Q&A. So Willem, Dave, Sue. Okay, so there are a few questions that have come up. The first one is at Dave, Prof. Berger. Are you investigating the use of hyperspectral imaging for auto ID? And he follows that up with, what research is being done on developing spectral indices for different crop or tree species?
Okay, I've unmuted myself. Thank you very much, Bernard. There was fascinating presentations after mine. And thank you, Reinhard, for that question. I know you also engaged yesterday about using hyperspectral imaging instead of just visual images. So, of course, what I was talking about was just using RGB, red, green, blue wavelengths or colors in your images. And so clearly hyperspectral imaging, where you're using different wavelengths, has got a lot of potential. And in FABI, there is actually a hyperspectral camera which we've got. And Reinhard, please send me an email and I'll put you in contact with those working on it. But I'm part of a project where we're already starting to look at some of these maize diseases using hyperspectral imaging. So we're very interested to work with you and to make sure the calibration is right and everything like that. I think it's got a lot of potential because you're looking at different characteristics of your images. Of course, the futuristic view is Bernard almost stole my thunder when he talked about the tricorder yesterday. And I'm sort of talking about an image tricorder kind of a tool, which might actually be fast, come on the market or be available quicker. And if we start using hyperspectral cameras, they're quite big and bulky, and obviously you've got to put them on drones, could be quite challenging. But there's definitely some work being done overseas on using hyperspectral cameras. There's a couple of publications we're looking at where people have looked at different pests and diseases. So we're quite excited to explore this field, and I think it's got good potential. So thanks for raising that, Reinhard.
Okay. Then there's another one at you, Dave, from Suresh Krishna. Prof. Dave, 75% accuracy can be taken into confidence if it is to be used by farmers who are not literate like scientists, but this approach is very promising. I guess it's more a comment than a question. Would you like to say something about the accuracy, 75%?
No, thanks for that comment. I think as biologists, we know that the world is not black and white, and there's always a lot of variability, and we're always going to have misidentifications, even with human experts. So we will never expect to get to 100%. So that's why we would like to kind of try to get this system that we're working out, trialing, once we feel we're confident that it's good enough. 75% is potentially good enough, but we would like to go a little bit higher to be able to have something which is reasonably reliable. But I think basically using an app, many people are very familiar with that. South Africa is actually quite different from Africa. In Uganda, there's more smartphones than people. There was a statistic I saw a while back, but they're not smartphones. But I think in South Africa, everyone's got smartphones. So even people who maybe are in rural areas would be able to use smartphones. So I think using this kind of just taking photographs and getting some kind of feedback, it could even be a verbal message could be quite powerful. Thanks.
Okay, great. And then there's one more for you, Dave, from an anonymous attendee who basically deleted it now. Okay, we'll leave it at that. Let's go to the questions on the chat. There was one to Willem. Let me just get it. Seems like these things have all been dismissed now. Yeah, one new message. Okay. Oh, sorry, it's to Candice. Has there ever been a case where insects or other agents involved in biocontrol measures have jumped to prey on other plants to detrimental effect? Or were the agents, did they have an impact on the ecosystem? Or where agents had an impact on the ecosystem?
Okay, thanks very much for that question. It's one we get quite often. Yeah, so we call those non-target effects, and we're very fortunate that in South Africa we have not had any non-target effects from any of the agents that we have actually introduced into South Africa for biocontrol. So a few steps back, a really important thing to know about the introduction of these agents is that they go through a really stringent process in terms of their testing, and this includes testing them in terms of whether or not they feed on the target plant that we are looking to control, or whether or not they reproduce on a target plant, for example. So Hackia, as an example, is a protea species. So when those agents were all introduced initially many years back before my time, they would have been tested against several protea species. And if there had been any indication that they favored or even did anything for that matter on an indigenous plant species, they would never have been cleared for release to begin with. So that is really important to know, that process is in place. So we have not had any negative non-target effects. It's not to say that we have had no non-target effects. And I say this because there is an agent which was introduced for the management of one acacia species, for example, which is now also utilized on another invasive acacia species. But both plants that it feeds on are invasive and are targets for management. So it has never jumped to, in South Africa, to an indigenous plant species that we are concerned with conserving, for example. But there is obviously globally the indication or the example of the cane toad in Australia, which was introduced for the purpose of biological control, and that went completely haywire. But it's a completely different system in that the cane toad is obviously predatory versus a highly specialized herbivorous insect species. So there's also a paper that might be of interest to you. I think it was Vilna that asked that question. And it was a paper by Harriet Hinds and colleagues in an opinion, insect opinion of science. I can share the link a bit later if you like. And that shows that less than 1% of insects that have been introduced for the purpose of biocontrol have had non-target impacts. And that's a global assessment. So it's very rare. Not to say that it doesn't happen, but it's getting better. So that 1% should actually be diminished as research improves.
Right, thank you. There's one more for you, Candice, from Johan Venter. Does the long lifespan of trees versus the short lifespan of insects and its exceptional adaptivity to surroundings not pose a problem when it changes or in future changes feeding and reproductive patterns to other species?
Okay, so I think that's a little bit of the same kind of question. So it wouldn't happen in that sense because, yes, trees have got a long lifespan and the insects might have a short lifespan, but just that alone means that the trees will be there for longer than what the insects will be there. So there's no reason for the insects to change their host. And it's just we were not looking in those timescales, basically. And these insects are really, and pathogens are really host specific. They are, it's like the monarch butterfly. I don't know if people know that example, that feeds only on milkweed, for example. So they're really, the insects can be quite fussy. So it's not something that is likely to happen.
Okay. Then there's one at Willem from Debbie Schultz. How much of the resistance breeding falls under genetically modified? If a significant amount, How do you deal with consumer perception? Is there a problem regarding perception?
So currently in wheat, there is no adoption of genetically modified resistant sources. There have been a lot of research in terms of gene cassettes and then also gene editing. So although these studies show a lot of promise, I think it is still regulated and not released in commercial cultivars as far as I'm known.
Okay. Thank you, Willem. Willem, I want to ask you a question regarding the use of hyperspectral technology and remote sensing in monitoring rust diseases in South Africa. I know that PANAR were playing around with this a couple of years ago. Has it advanced at all? Is anybody doing this?
So I think you referred to a study where it was used to screen some wheat lines. So it is still possible to do that. But in detection, then I think we have talked with Dave some time back to really start looking at that. So so then it can be included in a in-field type of detection. But from our research view, also it's important that we bring back isolates from the field to use in our studies to study relevance. So there are different issues here in terms of identification and then characterizing these isolates in terms of their virulence and avirulence profiles. But in resistance breeding, I think, yeah, definitely we will later on talk further with Dave. So see what we can do to optimize our screening in greenhouses at least, and see whether we can apply some of these tools in coming up with more accurate results here.
Okay, great. One more from Dennis Hunt to Candice. The control of hyacinth on the Hartbeespoort Dam to an outside observer seems not to have been effective. Any comments as to why?
Hi, Dennis. Yes, I'm reading the questions in the chat now. So that's a really good example of how programs are successful in some instances and not in others, and that there's other factors that confound whether or not it actually is successful or not. So Hartbeespoort Dam, it's not my area of research in particular, but I'm a bit familiar with it in terms of the high degree of nutrients that occur in that system, which obviously then leads to mass water hyacinth bodies, which just it becomes impossible for the insects to get on top of. So it's a complicated system, and that's what I hear all the time from the researchers at Rhodes University with regards to that system, and that high nutrification in the water is what actually is causing an impossible situation effectively. So I think, in short, that's as much as I can tell you on that front. Yeah, there were a few others there, Wynand. I don't know.
Okay, there's one that Sue is directing at you, Sue Walker, about the biocontrol of pinky weed in Gauteng. Has it been attempted?
Oh, yeah, so I was trying to find out what pinky weed is. I don't know. Is that the cannabis species? I've got no idea what pinky weed is. So a short answer to that would be no, because if I haven't heard of a biocontrol program being initiated against it, then probably not. But it is impossible. The capacity within the biocontrol fraternity, as much as there are many researchers, there are way too many weeds for us to get on top of. And it's obviously this, again, I guess, in terms of what the whole NSTF discussion group is about, is funding, and whether or not we can get funding to work on all of these different species. So someone there, is it pom-pom? Someone saying potentially pom-pom weed? Yes, no. Pom-pom.
Oh,
no.
Okay.
I wondered if it was pom-pom, but it's not pom-pom, and it's not Parthenium either. So pom-pom and Parthenium do have biocontrol programs on the go against them, but something called pinky weed I'm not really familiar with.
Okay, Candice, then there's a comment from Mike Wingfield. Mike says, thanks a lot for a very good summary of weed biocontrol in South Africa. And he asks, when introducing a new biological control pest, how much attention are you able to give to introducing a sufficiently broad population, brackets genetically, in order to avoid genetic bottlenecks in the longer term?
Yeah, thanks, Mike. That's actually a really good question. And I think it depends on the situation that you're working on and the system that you're working on and how quickly species are able to turn over and how much reproductive output they actually have. So I know there's no real answer to that question that we can apply to absolutely every single system that we work on. It's sort of like asking how long is a piece of string. But I know from my ecological background and my training, I obviously would try to introduce several thousand insects from varying populations, if at all possible. And that is one of the things that actually could impact the success or failure of a program. The genetic bottlenecks, perhaps, but also collecting something from one site, which might be really different to the target sites in the introduced range. So it depends entirely on how much time you've got, what system you're working on. And I guess how reproductively viable or how many generations a species might have in a year as well, for example. So I've collected the stem-boring beetle for Hakea, and I made sure that I collected several hundred each time I went to Australia or came to Australia to collect them, and my predecessor, Tony Gordon, did the same. I'm just looking at the chat now, and Sue, it is pom-pom weed that you're talking about. So yes, there is a biocontrol program against pom-pom weed that has been on the go.
Okay,
Mike, I hope that answers your question. Sorry.
Then there's one question from David Livingston. Prof. Berger, great talk. Does hyperspectral imaging take into account disease stages, especially for foliar diseases whose initial symptoms are indistinguishable?
Okay, thank you, David, one of my colleagues. So the question is about the potential of hyperspectral imaging for the different disease stages, and actually you hit the nail on the head here, that the images that I showed you were quite late disease symptoms with the RGB images, and often you want to be able to detect these things much earlier in the season. So that's really where I think hyperspectral imaging, but finding that specific wavelength band, which is going to work, which is going to pick up some kind of signal which represents kind of an early infection. I think that's where the exciting developments are. So we will clearly have to start with that type of studies in controlled glasshouse trials, like we're doing with some of our diseases that we've got nice systems going, and like the rust diseases that Willem was showing, the beautiful glasshouse trials that he can do there. So if you start doing artificial inoculations and start to look at that with a hyperspectral camera, that's definitely what we'll be doing. And I think that's the way we can start. And, we heard about early warning systems in the bigger idea, but actually this is an early warning system in a farmer's field. And so I think there is a lot of potential for that. So thanks for that question.
Okay, and then I think there's time for one last question. Actually, Dave, you directing it at Candace. Can members of the public release biocontrol agents? How is it regulated?
Thanks, Dave. So that's actually a great question, because I enjoyed giving this talk because there was a lot of talk about community engagement and citizen science. So they can, but they shouldn't do it without informing the required authorities. And the reason for this is obviously because it's linked quite strongly to long-term monitoring questions for research. So if people are just collecting and redistributing agents without informing somebody, if I were to go and look at something in terms of how well a species distributes, and I didn't make that release myself or know somebody that did, I would automatically assume that that insect has gotten there by itself, which obviously then skews our data quite significantly. There is work that I have spoken with recently with Crew down in the Western Cape, which works with the Botanical Society, and they are quite keen to get involved in redistributing biocontrol agents. There is something, mycoherbicide on Hakea, which we very happily redistribute to farmers or to other stakeholders, and we give them directions as to how they can actually do this themselves to manage trachea on their property, because it's a slightly different system to that of the insects, for example. So yes and no. Yeah, it's not impossible, but it just has to be controlled and managed. We don't just want people to go and throw things out without informing the person who's responsible for that program.
Okay, there was one question. Dave, you want to comment?
Yes, thanks, Candace. So if there's members of the public listening, where do they get hold of these biocontrol agents if they specifically wanted to do this? Who do they contact?
Yeah, it's really hard. So Department of Environmental Affairs has got what they call biosecurity officers in every province. Some of them work better than others. Some of them are more easily contactable than others. And effectively, that is the person that you should be liaising with, because that person liaises with the researchers across all organizations. However, I dealt directly with farmers myself and just made sure that this was listed on a overall database that we designed myself while I was working at the ARC. But it's not the easiest thing. So I would suggest contacting the researcher who's involved with that weed. And that, again, it's spread all over the country. Waterweeds, if it's a waterweed, I suggest looking at somebody at Rhodes University. They usually do that. Cacti species is also predominantly Rhodes University. A lot of tree species and shrubs are done through the ARC. Wits University also does some research on that. So it's easy enough to find the people, and the people who are actually doing the research on the programs are really contactable quite easily. It's just whether or not they themselves will be able to help you with the agents is a different story, because obviously, as I said, some of the agents are mass reared, others are field collected, which is why I had quite a big hand in terms of implementation and redistribution of agents, because my agents were field collected. But when it comes to mass rearing, it's a slightly different thing, and there's more players involved. But yeah, I would suggest contacting Rhodes University. Philip Ivey is now supposedly, I think he's got multiple different hats that he wears, and one of them would obviously be liaison with a lot of different stakeholders.
Wow. Okay. There was one I missed, Dave, at you from Jan-Hendrik Venter. Sorry, and I promise this will be the last one. He says this is a great tool to use for pests not yet in South Africa. Is there, for instance, some work done to identify virus symptoms which may contribute to maize lethal necrosis?
Yes, so obviously maize lethal necrosis is this big threat which is in East Africa, and it's a combination of two viruses, a potivirus and a maize chlorotic mottle virus, I think, which have their own individual symptoms. And obviously there are scientists already pre-screening. I was helping out with one of those surveys to see, we already have lots of potiviruses in our maize, but we don't have the other virus, so it's not here yet. And this can answer the other question which disappeared, that whether we are looking at other diseases. Clearly we can also, the next phase of the development will to have images of maize streak virus at different stages and other potential viruses that we can actually get images from overseas maybe and incorporate it in this. So it could be used for that kind of detection, so that if it's implemented and something comes in, maybe a member of public will be the first one up in Limpopo or near the eastern, the Kruger Park will be the first one to see the evidence, and if that can be got to the authorities quickly, then that would be really useful. So looking at virus diseases is definitely something which has got potential. There's also a lot of next generation sequencing work being done, screening plant material for the presence of viruses. We ourselves actually, by serendipity, found a novel virus. It's a harmless virus in maize. It's distributed around the world, and it doesn't seem to have any major effect. But that was something which we reported to Jan-Hendrik Venter as soon as we found that, so that he could know. And we've published that work now, so that's also a part of evidence-based data. So next generation sequencing is also a really powerful tool of finding these kind of pests and pathogens. Thanks.
Okay, and then I promise this is the last one. I don't want to, pour water on the enthusiasm. One doesn't get this level of participation and enthusiasm at live congresses. So apologies, Jansi, we're running a little bit over time, but this one just comes from Dennis Hunt, and he says, one wonders whether there might be room for a solution to wood borer to be engineered between the Fabi and Candice and her people. Any comment, Candice? You don't need to.
I'm not really sure about engineering insects. Yeah, look, that's my favorite little insect, to be honest with you. I don't think we need to engineer it. I think it does a really good job on its own. And yeah, we actually use some new technology such as CT scanning before I left the ARC to try and determine how well it was actually doing, because we were a bit worried about it dying in the field, and it's 100% survival. It really develops really well. So I'm not sure we need to genetically engineer it, but yeah, there's always room for work together, but I'm not sure in that aspect.
Okay, then I guess let's wrap that up. Thank you very much, Candice, Dave, Willem, Sue, for excellent presentations. I think the level of discussion that they generated is bears out, the quality of your presentation. So without further ado, then, Jansi, over to you.
Thank you, Wynand, and thank you to all the speakers. This was absolutely fascinating for the second day in a row, and all the presentations. They were just so intriguing and thought-provoking, as well as informative. Right, what's going to happen next is that presentations and videos of the speakers presenting will be on the website within a week, and those are with the speakers' permission. So please, speakers, if you've agreed to share your presentations, please send them to us, send them to Gabriella, so that we can post those on the website, obviously in PDF form. Complete proceedings will be ready in 3 weeks' time. They are being done by Right Connection. Cynthia's doing that. And then there is a media release being produced by Debbie Schultz, and she will finish that as soon as possible, but it could be 2 to 3 weeks. Please look out for that. That is the best way you can actually remind yourself of what was being discussed here. It's a good way to see what kind of issues came out, so that there can be possible further discussion or further action. And then please have a look, when you're on the website, have a look at our previous 2 discussion forums, preparing for epidemics in South Africa, human and animal. There were some interesting things that were similar in those presentations. The role of data specifically, the role of new technologies, and the importance of genetics, of course, and expertise in South Africa, mathematical modeling, and using cutting-edge technologies. It was also said that really one shouldn't make these demarcations watertight, that if you're looking at humans and animals, you should actually consider the plant health as well. So this can be read as a follow-up to the Preparing for Epidemics discussion forum. Last year, we had a seminar on Internet of Things technologies and 5G rollout. Have a look at that as well. There were 2 speakers there, and that is interesting for the high tech that we are having to use in our control of epidemics. Right, then, I want to thank the team, Dave Berger, Wynand Swart, and Ansa van Vuren, for co-chairing, and also for being the core of the team that conceptualized this discussion forum, for identifying of speakers, and for your incredible enthusiasm that made this work and made it work well. Also to Mike Wingfield and Mariana Purnell for their input into the conceptualization of this discussion forum. With that, I'm going to close off and wish you a very good afternoon and evening. Stay warm, stay safe, watch out for the wave that we're in, and we'll see you at the next discussion forum, I hope.